And these are the real figures of for the final analysis of metatranscriptomics data. For example, this is figure number 1, which is basically giving the details from pipeline to the gene expression level and comparative gene expression patterns in different types of samples. For example, C and D1, they are sample names. Then 24.6, 24.0, these are the sample namings. So there are different samples, and all these samples have been compared for the gene expression values under different conditions: hypoxic, non-hypoxic, so under, or you can say under stress condition. And these are the gene expression values in 2 samples for this particular genes. So these are the names of genes. For 80 is basically Arabidopsis thaliana, you can say this thing. And then for most of the top ranked genes, we have plotted the heat maps for the expression values. Now, if you want to go for functional analysis, then there is possibility that you can go for functional analysis, and these could be plotted in this way. So these are the KEGG pathways for the differentially expressed genes, up and down regulated genes, green algae. And then you can see complete details for all these samples. So this is basically the details which you might be interested how to plot and present the metatranscriptomics data. Now, if you are interested in deep exploration and understanding of your experiments or project, then you could go for some co-expression network analysis for the best or top differentially expressed genes. For example, these are the differentially expressed genes. Basically, these are the functional network for the differentially expressed genes. So, but these are based on the differentially expressed genes, and these are the networks. And this is quite different things. For example, you can say pathway crosstalks in these algae or the samples. I am not going to explain the details because this is somehow associated with someone's project, but this is for the help for those people who cannot go for training, paid training. This is for free learning for anyone from any country, any city, any place. Okay, so this is the best way. By the way, here you will need another tool, that is cytoscopes software, which will help you to plot these kind of networks. Now, there are some other networks, and these are all for co-expressed genes and their functional inferences in green algae for different time periods. So the samples were kept under stress, for example, for 24 hours, for 48 hours, for 72 hours. Now you can go for more details, and this is now for the gene co-expressed genes. And the same thing have been plotted here as a big and combined networks, and these are clustered genes. So the clustered genes and these links are showing the interaction between different up and down regulated genes, and the thicker length and higher deep color for red means a strong co-expression values, or you can explain it in your own way. So now you can also classify them for the biological functions, and you can see the details like the red node is representing up-regulated genes, blue node is representing down-regulated genes, and red line means positive correlation, blue line means negative correlation. Now go for better, like a screened network from this complex network, then it could look like this. For example, this is one of the gene which is connected here, but they are connecting with larger number of genes. Thus, you can say these genes are kind of master regulators. So you can learn from this video how to screen out minimal number of genes and with higher significance from the complex network of the co-expressed genes or in any kind of gene networks. And these are complex networks, which is not that much clear to understand. So you can go and try to screen the network based on the correlation values or some other parameters which you could be interested for your project. Now, if you want to present these figures in more beautiful way, then this is how you could also present the same research in another way with more information. And these are basically showing the bigger nodes means higher significant or smaller nodes means higher significant. So this could be opposite, but you need to see the videos properly. For example, in this case, it is opposite. These small nodes have higher connectivities. That means they are more significant. And bigger nodes are having lesser connectivity. That means these genes are having less significant. Or if we are talking about pathways, then the same thing applies for the pathways. Now, if you want to present the gene expression heat maps for the best genes in different samples, so here you could see these samples are present here, then the expression patterns for the identified genes in different samples for different time points and in different conditions. So you could look over your data and goal of the project and finally proceed for the real analysis. And it could take few weeks for completion of analysis work, but you need to spend the time and try to think how to best present the figures and interpret them in order to convince the reviewers and editors for higher quality research paper publication. Finally, we could conclude that it is metatranscriptomics is a powerful approach to study gene expression patterns in a microbial community selected or multiple microbial communities. It enables the researcher to understand the functional dynamics of a complex ecosystem. And finally, you are done with this. By the way, there are some people who try to give you the message that to analyze or to perform metatranscriptomics is complicated task, but it is as easy as RNA-seq analysis is being performed. So there is no difficulty. Only one thing what you need to know to understand the pipeline of metatranscriptomics and metagenomics and RNA-seq analysis for a well-defined organism, for example, human being. Then you will really understand and immediately perform the analysis if you have really good configuration computer. And that's all. Thank you.